Okay, welcome everyone. Welcome to Bodega Marine Laboratory and the inaugural launch of the Donald Michaels Distinguished Lecture Series. We're really excited to see all of you here today, and thank you for joining us today. My name is Eric Sanford. I'm a professor of evolution ecology who's based here at Bodega Marine Lab, and I'm also serving as the faculty host for Dr. Pespeni's visit. This new lecture series has been several years in planning, and it began when Don's wife, Vicki, and son, Christopher, approached then BML director, Gary Chair, with the idea of funding a project that would both benefit BML and the surrounding community, as well as honor Don's scientific career. And the goal of this new summer lecture series is to showcase the research of distinguished scientists who integrate physiological processes across levels of biological organization and disciplines to address grand challenges in marine and coastal sciences. And so the resulting week-long event begins this morning with a research seminar. On Wednesday, we'll have a community lecture that you're all invited to, which will provide a broadly accessible overview of Dr. Pespeni's research. And then on Thursday afternoon, we're also going to have a workshop where Dr. Pespeni plans to lead a discussion about ways that we can increase diversity and inclusion in the marine sciences. And then in addition, throughout this week, there will be lots of opportunities for students and other community members to benefit from informal meetings and social events with Melissa. So many of you are already familiar with Don Michaels' deep connections to BML. In the mid-1970s, Don began his PhD studies at UC Berkeley here at Bodega Marine Lab. And his faculty advisors and mentors included Cadet Hand, Ralph Smith, and Howard Byrne, all of whom were instrumental in the establishing and founding of BML. Don was a Sea Grant trainee, and his dissertation research focused on molting and lobsters and Dungeness crabs. Don earned his PhD in 1979 from UC Berkeley, and then went on to do a postdoc at Tennessee's Oak Ridge National Laboratory, where he studied the Caribbean land crab. And then in 1985, Don accepted a faculty position at Colorado State University, where he established the Crab Lab. And in 1998, he spent a sabbatical year here at BML with Dr. Ernie Chang, beginning a nearly 20-year collaboration on various aspects of crustacean physiology. During this time, Don brought many CSU students to BML for research opportunities. And as many of you know, he continues to be a familiar face here at BML, visiting several times a year as he pursues his ongoing research on invasive green crabs. So Don's had a very successful research career with major research grants from Sea Grant, the NIH, NSF, and other sources. He's internationally respected as an expert in the field of crustacean molecular and cellular molting physiology. He was a Fulbright scholar, he's a AAAS fellow, and he's published numerous scientific papers in Journal of Experimental Biology, Comparative Biochemistry and Physiology, and many other top journals. And many of these papers report results from research that was conducted here at BML. So this new lecture series is really a fitting tribute to Don's nearly 50-year association with Bodega Marine Laboratory. BML is very grateful that this new Distinguished Lecture Series is funded through a generous continuing gift from Vicki and Christopher Michaels. They invite you to consider joining them as partners and making a gift of any size to this new program. And our hope is that working together, we can make this lecture series available for future marine and coastal scientists in the broader community for years to come. And you can find a link to donate at this website on the screen, also in the email that many of you received announcing this event. Okay, so now it's my pleasure to introduce Dr. Melissa Pespeni from the University of Vermont. Dr. Pespeni is a creative and dynamic evolutionary biologist who applies cutting-edge genomic, genetic, and computational tools to address really some of the most important and timely challenges in biology today. In particular, much of her research asks the question, to what extent can marine species adapt to climate change and diseases? Dr. Pespeni received her PhD from Stanford University in 2010, where Steve Palumbi was her advisor. And her dissertation addressed whether purple sea urchin populations along the coast are locally adapted to environmental variation along the West Coast. And when Melissa began this work, really the prevailing wisdom was that species that had broad larval dispersal, like purple sea urchins, were thought to be essentially genetically homogeneous along the whole coast, basically from Mexico to Alaska. But using powerful new genetic approaches, Melissa was able to detect previously hidden signals of natural selection on purple urchin populations along the coast. And her dissertation was really influential because it was among the first to apply these new genomic methods to reveal that there's actually a lot more selection and population differentiation along the coast than had generally been appreciated. So after receiving her PhD, Dr. Pespini stayed on at Stanford as a postdoc. During this time, I had the pleasure of working with Melissa on a collaborative project with our Bodega Ocean Acidification Research Group here at BML. In particular, Melissa led a project to test whether sea urchins have the capacity to adapt to ocean acidification. So we cultured sea urchin larvae here at BML under different pH levels, and Melissa used sequence data to uncover these striking shifts across the genome in response to selection imposed by acidified conditions. Then Dr. Pespini further expanded her range of interests and approaches during a second postdoc at Indiana University. And here she investigated the evolution of novel traits in horned beetles, which extended her skills in genomics and genetics into the realm of developmental biology. This work was obviously quite distinct from a lot of her earlier work in marine systems, and I think it really shows that Melissa's drawn to pursuing important questions in evolutionary biology wherever they might take her. And then Dr. Pespini was hired as a faculty member at the University of Vermont in 2014. And over the past 9 years, she's established a really dynamic research program at UVM that's focused on climate change adaptation in coastal marine ecosystems. She received a prestigious NSF Career Award and has been using innovative selection experiments along with genomics to study the scope of adaptation within marine populations. And I'm continually impressed by Melissa's really novel and ambitious experiments, some of which you'll hear about today. For example, recent studies of experimental evolution from Melissa's lab involved raising copepods for 25 generations under factorial combinations of temperature and ocean acidification. And the results of that work have been published over the last few years in a really impressive series of papers in Nature Climate Change, Nature Communications, and PNAS. I think these are some of the most elegant and comprehensive studies of adaptation to climate change that I've seen published. And the work has really further cemented Dr. Pespini's reputation as really an international leader in studying adaptation to climate change. And given the influence and impact of her research, it's no surprise that she's in high demand as a speaker. So she's giving keynote addresses and invited seminars in Canada, France, Spain, Australia, Japan, Finland, Sweden, and top universities across the US. So we're really fortunate to have her here with us this week. I'll just add that beyond her stellar research achievements, Dr. Pespini has been really deeply committed to teaching and mentoring. For the past 6 years, she's been the director of an innovative graduate training program at UVM called Quest. She also has worked very actively to advance diversity, equity, and inclusion through mentoring, workshops, and a variety of other efforts, both at UVM and beyond. So we're really thrilled to welcome Dr. Pespini here at BML for the first Michaels Distinguished Lecture. And with that, I will turn it over to Melissa.
I think so. If I could do all the things you just listed, hopefully I could. Hopefully. Oh, I think that it's showing. Okay. Okay, can everybody hear me all right? Yeah? Okay, everyone in the back? All right. It's so wonderful to be here. Oh my gosh, that introduction was amazing. Thank you so much, Eric. And thank you all for coming out for this seminar and for this invitation. Such an honor. Thank you so much, Vicky and John, for making such a special event. Thanks to Eric and Emily for the nomination and the committee for the selection, and for Jennifer, Jessica, a number of people for all the details for actually getting me here. So thanks for them. So, okay, yeah. How about that? How's that online? Sound is a little muffled. Hard to hear. Is it better? Is it any better? Testing, testing. Is it any better for the folks online? Is it a little bit? Okay. I can put the same mic that you used. Do people not use the lapel mic much? Or sometimes. All right. No, it's okay. Is that better online? Yeah. Okay. All right. I'll try to stay behind the podium. Okay. Here we are being favorite. So you'll get to hear this one blue marble. I could show you. I think every single person. So if you feel comfortable, close your eyes. Picture some of the variation you know about that organism, either between individuals or species, related species. It could be something morphological, or it could be something physiological, special that you know about that. So relish in that. Not a moment. And bat your eyes open. It's that incredible variation that I want to study. We in my lab group love to study, and we want to understand how it can in incredible organisms. We primarily sea urchins, sea stars, arthropods, and we like to understand that diversity, how they adapt to local environment, how those mechanisms might their capacity to. And we've all probably phenotype. This graphic model has really exciting genomics. We can understand organisms to adapt. And we can also, in this crazy time where we've got an N of 1 experiment, and the relationship between the genotype, phenotype, and the environment is much more complex because of the role of microorganisms, for example, in affecting this relationship. But also because of epigenetic. Yeah, John. Yeah, oh yeah. That's okay. I can just hold it. Okay, now I'm more free. All right. Okay. So yes, we have this more complex relationship than we ever thought was possible before, right? So the relationship between the environment and the genotype can affect the phenotype based on the microbes that live in and on an organism, but also these epigenetic marks that we used to think didn't necessarily play a role. So these are some of the exciting frontiers. And lastly, we can validate these relationships in ways that are unprecedented, using techniques like CRISPR-Cas9 or other things to validate this relationship. So I'll tell you 3 stories today. One about purple urchins and how populations can adapt to local pH variation in the wild. Another about copepods and if they can adapt to these future global change conditions using lab experimental evolution. And then lastly, I'm not totally going to answer this question. It's a disclaimer. So what's the cause of sea star wasting disease? But we'll get at some potential, some implications at least of what could be causing it. So the purple urchin, which is, I guess, kind of a nemesis around here these days, particularly with the loss of their predators, but they're this broadly distributed marine invertebrate that lives in intertidal and subtidal areas from the cold waters of Alaska all the way to the warmer waters of Baja California, Mexico. And they can disperse in ways that make them a really robust model for evolution studies because they have high gene flow up and down this broad latitudinal gradient. And they've also been a model for developmental biology for centuries, and in the last 20 years have had their genome sequenced. And so much is now known about the function of many different genes in their genomes. And a student, Jenga Patek in my lab, wanted to go a little deeper in this understanding, and particularly not just mean conditions, average conditions along the species range, but the degree of variability, because she's really interested in the evolution of gene regulation. So she wanted to find populations that had consistently more variability in pH, and bodega here is one of those, where the pH more frequently gets down to really low conditions, in addition to Fogarty Creek and Cape Blanco. So she was interested in that variation and sampled 7 populations with the help of Eric and his lab, as well as many other folks up and down this range. These samples were collected during the pandemic, so it was really interesting challenge, and sequenced their whole genome. So 140 genomes. And across all those 140 genomes, identified about 1 million single nucleotide polymorphisms, so genetic variants across the genome. And this is a principal component plot, which is a way to partition variation of any set of data, but in this case, we're looking at genetic data, across your set of samples. So in this case, we have 20 individuals from each of these 7 populations. Each of these dots represents an individual, and each color represents one of these different populations here. And what you can see here is a complete mess. There's no clustering by population, which is what we actually wanted to see, is that most of the genome, based on what you would expect based on the dispersal ability of these organisms, is completely shared. So this genetic variation is shared across the species range. And there's tons of genetic variation. So we see high genetic diversity and no population structure, which makes them a robust model against which we can look for signatures of natural selection. And so this is looking across the genome all lined up at this genetic variation. And here on the right side, you can see this histogram. Most of the genetic variation is really low diversity. So this is looking at the degree of genetic differentiation between populations that experience high pH variability and those that experience low pH variability. And you can see that there is this distribution of about 10,000 SNPs evenly distributed across the genome, which those are the ones that are associated with pH variability. But she wanted to know where they are across the genome and what types of genes. So there is a signature of adaptation to pH variability, but it's polygenic, meaning many genes involved. We're not seeing like a single spike. If you've ever looked at say, coat-colored polyphenisms or things like that that are, have a simple genetic basis, you'll see a large spike. But when you're adapting to pH or temperature, you're going to have a broad distribution of genetic variation. But we wanted to see if it might be clustered in other ways. And one way to do that is to look at the types of regions in the genome where that genetic variation might lie. So in this case, we have characterized all the genetic variants as to whether they fall in introns or exons or in untranslated regions or an enhancer or long non-coding RNAs. And you can see, looking at the observed versus expected number of polymorphisms, that these enhancer and long non-coding RNAs, which are regulatory molecules and regions, are dramatically overrepresented in terms of variants that are associated with pH variability. So they're in regulatory regions, but they're also, when we look at the network of interacting proteins, they are distributed across the network. So this is a random subset of some 1,000 proteins and how they interact with each other. The bigger dots are the ones that are the significant outliers that are associated with pH variability. And you can see that they're not central in these networks of interacting proteins, which makes sense. You're not going to mess up things that are core to your survival or to your functioning. So if you think about hubs and spokes, it's at the end of the spokes where this adaptive genetic variation might lie. So it's on the periphery of these protein-protein interaction networks. And one particularly interesting set of genes where you can have this sort of, where you might expect to see a signature of selection to pH variability is in larval skeletal genes, or in skeletal genes for urchins in general. You're looking at a Puteus larva here, and you can see their gorgeous biomineralized skeletons with these rods here that are covered with cilia, and that's how they can, the cilia beat, and that's how they feed and are able to swim around. When conditions, pH conditions lower, it's much harder for any calcifying marine invertebrate or marine organism to deposit that calcification and grow. So this whole suite of marine or biomineralization genes is important for understanding capacity to adapt. So if we look at all SNPs and build a phylogenetic tree and characterize each site, each of these 7 sites, based on the degree of variability in pH, you can see that across all those genetic variants, there's no clustering across this phylogenetic tree. But if we look at just the all the SNPs that are in the biomineralization genes, you start to see some clustering by the degree of pH variability. And if you further look by just those outlier SNPs that are in the biomineralization genes, you see this nice gradient that's organized by the degree of pH variability that you would expect, which goes in line with the experiments that Eric mentioned that we did many years ago, where we showed that larval survival really depends on the genetic variation that exists in these biomineralization genes. So we see that again here. So the local adaptation is likely happening through selection on dispersing larvae, and in part in these genes. And if we look at a handful of these biomineralization genes along these sites organized from San Diego to Fogarty Creek, and look at the allele frequency, and in the black line is the pH variability, you can see a general correlation of some of these here. And for a population that has that lack of genetic structure that you saw in that first PCA that I showed you, this is an incredible shift in allele frequency. So some of these are an allele frequency change of 20 to 40%, which is huge for sea urchin. And how do they do this? Well, I alluded to that before. It's through selection, balancing selection on dispersing larvae, and variation, balancing selection, maintaining this adaptive genetic variation across these populations. And we want to validate these. So another thing that Chenga is doing right now is green fluorescent reporter assays, where she's putting in some of the mutations that are upstream of particular genes of interest into reporters and injecting them into the sea urchin embryos and looking at the magnitude of expression and testing specifically how do these regulatory mutations affect gene expression, and does that change depending on the pH conditions that they're in, or does that change depending on the genomic background that they have, were they a San Diego urchin or an Oregon urchin? So more on that, and this is also, I have a postdoc position available in my lab for folks that are interested in doing this kind of molecular genetic genotype to phenotype work. So yes, the sea urchins can adapt to pH variability, and they do so largely through regulatory, peripheral, and also biomineralization genes. But that was just, these are experiments that nature has done for us, so that selection has happened in the natural world, and we're getting an idea based on the variability in the natural world using sites like Bodega that experience low pH conditions more frequently as a proxy for what might happen in the future. What we can do with copepods, because they're these short-lived organisms, is do this experimental evolution in the lab and test whether or not they can adapt to these future global change conditions. Acarcia tonsa is the organism that we primarily work with, and one of the reasons that I love them is because of this egg to adult, reproductive adult life cycle can happen in just 20 days, but also they are ecologically foundational. They are the bottom, the baseline, or the base of the food chain, this link between the primary producers and the primary consumers in the marine ecosystem. And they're also important for biogeochemical cycling, so sequestration of carbon and sinking of that carbon. And actually, I'll talk more about some of these results in the lecture on Wednesday, the community lecture. But for now, I'll tell you about how we experimentally evolved them in this full factorial experiment where they're reared in ambient conditions for AM there. Ocean acidification alone, ocean warming alone, or the combination ocean warming and acidification. And you can see we measured egg production, hatching success, survival, etc. It integrated all of that into population fitness, as you can see here. And after just one generation in these conditions, ocean warming and acidification animals in the 4 replicates that were in those conditions dramatically declined, the fitness declined, but recovered after just 3 generations, miraculously. And then everything was stable until about 25 generations, and there was a decline there. And this is work that was done in collaboration with Hans Daum's lab at the University of Connecticut, and really led by Jimmy DeMaio as the grad student carrying out the experiments. And we wanted to look at what was happening on the genetic side of that. So Reed Brennan, former postdoc in my lab, now junior faculty member at GeoMar, sequenced these 4 replicates times these 4 conditions and the founding population, and partitioned that genetic variation across all of these samples, again using a principal components analysis. What you can see is a strong signature of genetic divergence, primarily between the founding population. We were looking at the axis that explains most of that variation, so about 16%, between founding and the ocean warming and acidification and warming, but also a signature of lab adaptation. So difference between, on the second axis, the founding and the ambient condition, as well as the acidification. So you see this really tight clustering based on the treatment condition that they were in, which is strongly suggestive of directional selection. And we also did a number of other analyses to show that warming was in fact the driving force, even in this combined treatment, but there was a unique synergy of having experiencing both warming and acidification together, whereas acidification alone hardly had any effect, even though the conditions were going from 400 parts per million to 2,000 parts per million PCO2. and the warming was 4 degrees, so from 18 to 22. We also looked at just overall genetic diversity. Was there a decline? So you think typically rapid adaptation, there has to be a decline in genetic diversity. So here is just an image showing standing genetic diversity, and it doesn't decline after they go through this ring of fire of adaptation. So they still have evolutionary fodder, perhaps, despite this long-term adaptation. But we wanted to know, are there costs? Are there other hidden costs? So we did what's called a reciprocal transplant in the middle of this experimental evolution. So after 20 generations, just focusing on 2 of the treatment environments, so ambient and ocean warming and acidification. After 20 generations, we put the ambient animals into the ocean warming and acidification and returned the ocean warming and acidification animals to ambient conditions. And then for each of 3 generations, sampled them and measured their gene expression and their allele frequencies, so their genetic variation. And looking just at the gene expression variation, at that first generation, you can see strong clustering by treatment. So the lines are shown by the shape. So if it was originally an ambient line, it's in a circle. If it was originally an ocean warming and acidification selected animal, it's in a triangle. And then the color tells you the treatment environment at that moment. So here are our original ambient animals in the blue circles, and our original ocean warming and acidification animals in the red triangles. And you can see that they're very different. Now we're partitioning gene expression variation here of some 24,000 genes. And you can see that there is a big change when these ambient animals are moved into the ocean warming and acidification environment. But for these animals going from the ocean warming and acidification back into ambient, there's less of a change, so less of a move here. Just using this this PCA space to show that, or to illustrate that here. So we see this strong clustering both by line and by treatment, and we see strong expression differentiation. So a lot of these types of genes were in response to stimuli, actin and cytoskeleton, so the structure of the cells, as well as development and maintaining homeostasis, cellular homeostasis. But by that 3rd generation, now we have strong clustering by the treatment. So both populations of animals that were transplanted came to change their gene expression patterns such that they matched the environment that they were put in over those 3 consecutive generations. So that it looks like what happened was that they readapted to that environment. And we looked at the allele frequencies and showed that indeed that was what happened. So the ocean warming and acidification animals readapted to those ambient environments, but they did so with a cost. They lost genetic diversity in the very genes that they had used to adapt to those ocean warming and acidification conditions to begin with. But I wanted to focus, I went over those sets of studies really quickly because I wanted to share with you some newer data that we're still trying to figure out. So wondering about an epigenetic response. So we did reduced bisulfite sequencing and identified about 96,000 variant loci across the genome, and did the same sort of partitioning of the variation to just see, it's just a wonderful way to see how similar your samples are. And again, you can see a signature of the founding population being different from the experimentally evolved. But again, more similarity between the ambient and acidification, meaning that there was less of an effect of acidification, but a strongest difference between the ocean warming and acidification and the founding populations, and a greater similarity between the warming and the ocean warming and acidification, showing that warming is really the driving effect, even though, and acidification is a minor consequence, even though it's a big change. So we see this signature. We wanted to know, which shows this clustering of replicates, directional changes, and that warming is the driving force and the synergy of the combination. So similar patterns that we saw with the genetic allelic changes. We wanted to see, are these happening in the same regions of the genome? Here's one way that we looked at it among many. So we partitioned all of the methylation changes as those that were significantly changing versus those that were not, and then looked at the average genetic divergence in those regions of the genome using this metric of FST as a way of calculating genetic divergence. And while it doesn't look like that much of a difference, if we model it, we can see using permutation analyses. So here's the distribution that you would expect by random chance. Here in the red line is what we observe. Highly unlikely to get this difference by chance. So we see that we have about 2.5-fold lower FST in regions that have significant methylation divergence. So these genetic and epigenetic changes are actually happening in different regions of the genome, which is very fascinating. And we think of methylation as this mechanism for controlling gene expression, so when and where a gene is turned on. And if we look at this relationship, we use our gene expression data from that reciprocal transplant study that I showed you. If we look at the relationship between the change in methylation and the change in gene expression, we see overall a positive relationship. So as methylation increases, there's higher gene expression in the ocean warming and acidification. Or as methylation increases in ambient, there's higher gene expression in the ambient. So a positive relationship there, showing that epigenetic changes are associated with this divergence in baseline differences in gene expression, which is really cool to see in this long-term experimental evolution setting. And then one more pattern related to the gene expression is that because we did that transplant, we were able to see their acute plasticity, so how they quickly respond to a change in environment. So we characterized genes as being either non-plastic, so they didn't show a change when they were moved in environments, or plastic, they showed a major change. And then we looked at the percent methylation in that ocean warming environment. And we can see that the genes that were not plastic, the genes that were plastic that had that really acute response had a much lower percent methylation. So that suggesting that methylation is leading, combining it with the previous slide that I showed you, methylation is leading to long-term differences in gene expression and reducing the ability for genes to be plastic and respond acutely to changes in environment. So these dynamic, acutely responsive genes have low methylation. So putting this all together, we find evidence for both genetic and epigenetic changes, directional changes, through experimental evolution, and they affect different regions of the genome. And the divergence in methylation results in divergence in basal or steady state gene expression patterns. But the plastic genes, so the acutely responsive genes, have low methylation. So that is what we are learning about the molecular mechanisms about how copepods adapt. The good news is they have genetic variation for this. The bad news is that there are costs. So when you switch them into a new environment, there were dramatic declines in fitness and loss of genetic diversity. So they can adapt, but it's limited. And that there's an interesting, unique interplay or interaction between, or perhaps a lack of interaction between the genetic and epigenetic changes. So there's more to figure out there, but it's really fun to be getting at the mechanisms underlying this adaptation. So yes, both with epigenetic and genetic mechanisms, but with costs. So now for C-star wasting disease, something that may or may not have a genetic basis for being able to adapt to this. These images I will sometimes show when I'm giving talks, and a lot of times it's because people haven't heard of C-star wasting disease, but you all have heard of C-star wasting, so this is probably like a traumatic set of images for you to see, not so informative. But we know that it's one of the largest marine epidemics ever recorded due to the geographic extent from Baja California to Alaska, the more than 20 species affected, and the 100% mortality of some species at some sites. Hypoxic events have been implicated recently, but the exact cause is still unknown. So what we were interested in when I was just starting my lab, I had a postdoc join my lab who had a background in parasitology, and she was interested in combining our interests in echinoderms and trying to figure out sea star wasting disease and what could be affecting this tipping point. So why now has it become such a big issue? Because outbreaks had happened in the past, but never to this extent. And we were interested in particularly looking at the microbiome because microbes are known to be important in both health and disease. They've been studied extensively in human-related diseases, but also in other marine diseases like black band disease and corals. But we know from all of these examples, so black band disease, pneumonia, irritable bowel disease, or even allergies, many of these diseases or sets of symptoms can be caused by an imbalance in the microbiome. And they can be caused by many microbes, so you're not necessarily looking for just a single one to be the cause. So we did both a lab study where we did longitudinal sampling of the same sea stars as they progressed through wasting disease or remained healthy, and looked at their microbiome, their genetic variation, as well as their gene expression, but also did field sampling of Pycnopodia, the sunflower star that you see here. And we're lucky enough to be able to sample naive, exposed, and wasting animals. So presumably naive, there've been no evidence of wasting in the area yet, and it was just moving up into that area. So first, for the lab study of Pysas ocraceus, we had collected 37 healthy Pysas ocraceus from Monterey Bay, an area that had recovered from wasting disease but had been exposed to it. And the plan was that we were going to quarantine them for 2 weeks and then do an experimental infection. They arrived, it was overnight shipping, 17 hours. They were in the lab. They all looked healthy and great. We started the quarantine, and then animals started showing symptoms in the lab. And so the experiment started just based on whatever microbes they had coming in with them. And you can see the progression of symptoms from 0 through 5 here. So here's totally healthy, this is 2, so 2 lesions, 3 lesions, pulling arms apart, and dead. So you can see the symptom progression through the 2-week experiment here. So there were 8 sea stars that ended the experiment totally healthy, and the rest progressed through wasting disease. And looking at the microbiome, so this is a plot where we can look at the relative proportion of all of these different microbial taxa, including an important group called unknown, that has not been characterized in the marine world, which there's a lot of that. And I'll point out just 2 things. The decline of this, perhaps beneficial unknown microbes through symptom progression. So they started out as much more abundant, but then decline. And then a complete turnover, or not complete, but a major turnover event happens where there's an increase in putatively opportunistic microbes as symptoms progress. So we see this pattern, even though in the healthy animals, this is looking at the same kind of microbial composition through time of just the animals that stayed healthy. There's a bit of a change in days 3 and 6, but it returns by day 9, such that there's no significant differences between the community composition between days 0 and 9, or 12 and 15. So the animals that remained healthy have a very resilient microbiome, despite the fact that they're in these artificial lab conditions. But we wanted to look at their gene expression patterns to see if we might have some indication of what could be causing this. And what we see is that in the animals that stay healthy, they have much higher expression of genes related to everything that's in orange was more highly expressed in healthy animals. Everything in purple was more highly expressed in wasting. And this is just a dendrogram of functional categories of genes that were, as they were expressed through wasting disease. So in the healthy animals, you see a lot of cell-cell adhesion, cell regulation of cell proliferation, immune-related genes, so cytokine production. So we see much higher expression of pro-collagen genes, so genes that control their collagen system, and immune genes in the healthy animals. So they are, were all evenly exposed as they came into the lab. Some of them succumbed to wasting, whereas others didn't. Those that didn't maintained control of their catch collagen system and had a healthy responsive immune system. And surprisingly, we saw lower expression of immune genes in all the animals that were wasting. So they had lost control of their catch collagen system, had lower expression of those, and higher expression of collagenases, so genes that degrade collagen. And a lot of genes related to RNA processing and splicing and things like that, that if we look at the specific genes, a number of them have been implicated in neurodegenerative diseases in humans, which interestingly, the collagen system for echinoderms is neurologically controlled. So as you all have probably done, tried to pick up a sea urchin or a sea star and suddenly they stick. So, or for them to just be able to maintain that cozy position in the rock without expending their muscles, to do so, they activate this catch collagen system, which is neurologically controlled. So it seems that in wasting disease, they've lost neurological control of this system. And we also see evidence of hypoxia, so higher expression of this transcription factor, the hypoxia-inducible factor 1 alpha, as well as those collagenase genes higher in the wasting individuals. But the pro-collagen genes are all more highly expressed in the healthy animals. And we might be underpowered here, but we looked at about 100,000 single nucleotide polymorphisms across these 37 individuals and found no association with genetic variation between whether or not they wasted or stayed healthy. So at least with this set of animals, it doesn't look like there's genetic variation for surviving wasting. In the wild sunflower stars, we similarly see a major turnover in the microbiome composition. So if we look at the naive animals, so ones that were from sites where there have been no evidence of disease ever, they have this very simple microbiome composition. So this is just a random subset of 5 out of 20 individuals that we had sampled. But in sites, we had paired exposed and wasting individuals. So it was a given site that had individuals that were showing signs of wasting as well as individuals that looked healthy. And when we look at those, you can see that there starts to be a turnover in the microbiome in the exposed individuals that dramatically turns over in the wasting individuals, whole new sets of taxa, and a major increase in the Vibrio group. And that Vibrio group is particularly interesting. I'll come back to that. But what this pattern of increasing diversity, so here's some common ecological indices. So in alpha diversity, the Shannon index, you can see this increase in diversity is support for the dysbiosis hypothesis. So as disease progresses, there's just a proliferation of many different microbes through disease progression that we can see a similar pattern in the field as what we saw in the lab. So it's this what's been proposed as an Anna Karenina hypothesis that what kills you, there's many different ways to kill you, basically, not necessarily a single pathogen. And we see the same pattern looking at beta diversity, so who specifically is there as you can see in this plot here. Going back to that Vibrio pattern, you can see this is looking at one particular taxa. Here's the distribution of abundance of this particular Vibrio species in the naive animals. So even with just exposure, so they're not showing any signs of wasting yet, you see almost over a 1,200-fold increase, because this is a log 10 scale, in abundance of this particular Vibrio group that stays high once they're in the wasting. So this showing up, on exposure, but before signs of actual wasting, could be a smoking gun, or could be evidence of what's happening in their environment. And just one more PCA, because I wanted to show you one in every part of the talk, is this partitioning of variation across all of the individuals in this study. So each dot is an individual sea star sampled in the wild, and this is partitioning now their microbiome composition. You can see that there's tighter clustering of the naive with the exposed animals, even though these geographically are much more distant. Which is positive support that it's not a geographic signature, that it's actually a signature of the wasting disease that's turning over. And you can see that change in composition, change as animals progress through wasting disease. So to summarize for the sea stars, we present evidence that it looks like it's a dysbiosis disease, like many other diseases that we have in humans, but there could be a single cause potentially, or not necessarily a single cause, but there could be a role for Vibrio. We see evidence of hypoxia in both the lab and the wild, so maybe there are low oxygen conditions that are leading to this turnover and increase in anaerobic microbes that are proliferating and causing the loss of the catch collagen system. More studies are needed. And they seem to be able to resist wasting with an active immune system and control of their collagen systems. And maybe Vibrio is an important player, at least it's an important marker of something that's going on. So for wasting disease, what's the cause? I can't totally tell you, but low oxygen seems to be important. Microbial dysbiosis, maybe a role for Vibrio. I'm sorry that it's still a mystery, but I think we're making progress, and I know other groups are doing more studies on this. So in the broader context of what I've shown you related to this model of understanding this genotype-phenotype-environment relationship, I've shown you that we can associate genome-wide genetic variation with environmental variability even, not just mean conditions, to identify genetic variants that allow organisms to adapt to local conditions. And we're trying to take that next step to manipulate genetic variation and link it to phenotypic variation with these. With the copepods, we've shown you that we can experimentally evolve these organisms by exposing them to different environments and watch both the genetic and phenotypic changes happen through across many generations. We show a role for both genetic and epigenetic variation. We have a student who's looking at the impacts on the microbiome now, an undergraduate. And lastly, with sea star wasting disease, we sadly don't show any genetic variation that seems like it could be helping them, but we do see a strong potential role for the environment and also the microbiome in shaping their phenotypes, whether they succumb to wasting disease or stay healthy. So your big take-home messages for this talk are that there's regulatory variation that's important for adapting to environmental variability, and show that with the sea urchins, and this occurs through selection on dispersing larvae, among other things. We show that there's a role for both genetic and epigenetic variation in adapting to global change conditions. And lastly, that the microbiome likely plays an important role in both health and disease. We need to spend more time studying healthy organisms so we can get a hold on that unknown category to understand, what could those potentially putatively beneficial microbes be doing to help out, and continuing to do monitoring to understand what the specific changes are that are happening in the environment. With that, I'd like to thank all of you for your attention. Thank Dawn and Vicki specifically for creating this lecture series, and Emily and Eric for hosting me and setting up such a fabulous week. And the many members of my lab, past and present. I was only able to talk about a sliver of the work, but I have the folks highlighted in bold and in the picture frames who I focused on. If you want to hear some other work, like maybe Lauren Ashlock's work, you can come to the talk on Wednesday. I'll talk more about that. And with that, I'd like to take any questions, and thank you so much.
Hello. Melissa would be happy to take any questions from the audience. I will ask you to speak into the microphone so people online hear the question. So just raise your hand if you have a question.
Really cool talk. I really enjoyed that. So much work. I had a question about collagen being neuronally regulated. If you could talk about that, because I was totally unfamiliar with how that works, and anything you could say about that would be cool.
Yeah. So, okay, I'm gonna not start with the caveat because women scientists start to do that. So I'm gonna leave that out. So what I know about catch collagen being neuronally controlled. So, for example, if you look at an urchin spine, at the base of that spine, you have muscle holding it onto the test. And underneath that is the catch collagen, right underneath that muscle. And it is connected to their neurological system. So and whenever they want, they can turn it on and it just locks. So it's interconnected collagen with ossicles, etc., that can just get into this locked position such that the muscles can relax. More on the mechanism, I don't know if anybody else knows more about exactly how they do it, more of the genes involved. I don't know that, I think that most of the papers published on it are from the 70s, so no gene expression transcriptomics or anything like that of those specific tissues. But that's what and all echinoderms have it. So even sea cucumbers have it and do it. Anya?
Awesome talk. Super cool. I have lots of questions for all myself. That's one. So with the copepod work, you mentioned the loss of genetic diversity upon return to ambient of the ocean warming and one. Was that true for all of the different family lines, or was it just for the ones that did the reciprocal transplant, like back to ambient?
That's a great question. So for that reciprocal transplant, because it would have just gotten crazy, we only focused on the ambient and the ocean warming and acidification and did the swap with those. So we didn't do the warming alone or the acidification alone. But the ambient going into ocean warming and acidification didn't lose any diversity. So kind of the control, if you will, was totally fine. And what's even cooler about that control was that when we moved them, even though they had been in kind of constant chronic lab conditions of steady state pH and steady state temperature, they still had physiological plasticity to launch a gene expression response to that new environment. So even though they had been in this, lab condition where they presumably could have lost plasticity over the number of generations, they didn't. So it was not something about the lab that made the ambient or the ocean warming and acidification lose plasticity.
That's kind of crazy. I would expect the opposite, but I guess what I was thinking, what does this for variability environment? That's going from ambient at that like non-constant what that plus
Yeah, great question. There is, actually a current postdoc in my lab, Matt Sasaki, is working on understanding seasonal adaptation across, because rarely will you have constant conditions like this, right? So he's taking this out to the field essentially and measuring adaptation as it happens as the environment changes in the field. So sampling early in the spring and as it warms up and then as it cools down and looking for those seasonal changes in allele frequency. It'll be really interesting too to see, do we see the same thing, similar types of genes under selection? Do we see, so he's doing this in 20 sites from Canada to Florida. Do we see the same genetic variants responding across latitudes or not? I mean, of course, the environmental change is not as strong across those latitudes, but that's a part of the test, right? So we expect less change happening in Florida, much more change happening up north in Gulf of Maine and in Canada. Yeah.
Thanks, Melissa.
Thanks.
So, I'm actually just curious with the pie disaster. It seems like not as many people talk about how it's possible for them to recover from wasting seeds, you know, not necessarily progress to death. And so I'm just curious if you or anybody else, you know, has been looking at recovering individuals and following either genetic expression or microbiome, you know, for those individuals.
Yeah, that would be really interesting. You know, now that you mention it, so we had, there were a couple times, and we have the data are all published, but there were a couple times where an individual would go from a 0 to a 1 back to a 0. So they would have a lesion that would show, and then we'd go back and there was nothing. So they totally, I mean, this is the way sea stars, you know, this can just happen with sea stars, and this is also just the way that they die, even, you know, regardless of sea star wasting disease. This is just losing control of their catch collagen system. It's something that can happen when they're old and die, etc. We aren't doing any more lab studies with sea stars because it was only that one, and we just feel bad. There's no reason to take any more sea stars from the field. So to do something like that, you just need to be able to keep track of them in the field, and I'm not close enough to do that for doing those kinds of studies. But if other people would, and yeah, yeah, that would be great to do. Yeah, and in the field would be really powerful.
Hi, Melissa. I was wondering about the biomineralization work, and when you look at the highly variable far north population, in terms of the change that you see there, what is that? What is that? What are the implications of that on the actual spicules? Are they normal? Are they calcified in the same structure, the same way, or you know, what is the adaptation for? And I also wanted to ask if you cross southern males with those females.
Yeah, that's great. So for the last question, Chenga is doing some of that. So she's crossing northern and southern animals and doing that with her genetic manipulation. So if you have a northern mom or a northern dad, how does that affect the gene expression phenotypes? Or what does it actually mean for the biomineralization? So, you know, the biomineralization happens across all of these life stages. So in the developing larvae, but also as adults and growing adults. So for adult phenotypes, as part of my dissertation research, actually, we did a fun experiment where we just trimmed spines from animals that had been adults that had been in the common garden environment at Hopkins for 3 years. So they were, I think, or maybe Fogarty Creek animals and San Diego animals. And we had them in a common garden for 3 years. And then we trimmed all their spines because I had biomineralization gene expression data that showed higher gene expression in northern population urchins. And we found that indeed the northern population animals regrew the spines at a much faster rate than the San Diego animals, which was matching the gene expression data really well. So those are the only those are the adult morphological data that I have. And then for the larvae, from the experiment that Eric and I did together many years ago, we found that the larval growth was indeed stunted. Like the overall body length was shorter, the arms were shorter in the low pH environment, but they survived and were there. So it means that they were at least able to deposit that to do the act of biomineralization. So even though it was reduced, we had another study where we were looking at a carbonic anhydrase, which is an important molecule for this deposition. And we found some really interesting mutations that we were going to do some protein modeling and things with that, and link it to phenotype. But interestingly, the way that carbonic anhydrase, this particular one, works for purple urchins is it has this dramatically long low complexity domain that is impossible for anything to model or to create synthetically either. So it looks, and what it's used for, at least what's known, is that it kind of gets left there. The actual protein gets left in the calcium carbonate matrix and helps with the precipitation of the calcium carbonate. So there's something there that is harder to study any further, even though I was working with amazing protein biochemists. But there's more to be known. That's what I know.
So the question I have is regard to copepod experiment.
Mm-hmm.
You raising animal generation all life history stage,
Yeah.
correct?
Yep.
Do you have any idea about, is there a particular life stage which selection takes place? I understand your experiment to have like adult, right?
Yeah.
So you have to talk about that.
Yeah. Lauren, you want to take this one? So yes, this was, alleles and then also gene expression just at the adult stages, but of course, all stages went through the environments. We know actually from Lauren's data that so she did experiments looking at the sensitivity of nauplii, copepodites, and then the adults, and found that actually the copepodites were the most resilient in terms of temperature resilience. So the teenagers were fine, but the babies and the adults had much lower thermal tolerance. So it could be that those are more of the selective bottlenecks. Also from that actual experiment, some of the we didn't see, I think Jimmy didn't test any differences among the life history stages, but we saw that hatching success and survival, so ability to survive to a reproductive adult and the ability for the egg to actually hatch into a viable individual were the two traits that actually changed in response to selection. Number of eggs and I forget all the other things that he measured didn't change as much. Yeah.
Take one more question.
Hi, Melissa.
Hi, Lauren.
I have a question. So, looking at differences in the methylation and how there was lower FSD
in the more methylated areas, and also differences in gene expression with those areas of methylation. So I'm wondering, like, with these different strategies that the copepods have, is there any pattern of why they would use methylation versus gene expression versus difference in genetic variation? Like if environmental variability has anything to do with that? Yeah, just curious.
Yeah, that's great. Yeah, help me figure that out because we're writing this paper. Yeah, I mean, I think that the big take home, my I don't know, maybe it's a cop out, but is that, there's many different mechanisms when you're adapting to both high temperature and high CO2, these kind of really polygenic, or stressors that you would expect to have a polygenic response. And for the, I find the integration with the gene expression data to be really fascinating. The fact that you have higher methylation that leads to chronic differences in gene expression. So that is the mechanism. So if you want, like, if it's better to have X gene highly expressed, you do that with a change in methylation. If you need Y gene to be responsive to a change in environment, you keep it unmethylated. For the genetic changes, it could be that, those are kind of the classic things that we're used to hearing about. So it's a regulatory mutation that affects the binding of a transcription factor, or it's a mutation in the transcription factor that affects the gene expression, or that example of the carbonic anhydrase. So it's a mutation that affects the deposition of something. That's urchins, but same idea. Something that affects protein function would be the allelic changes. So I think that when we're talking about these complex phenotypes in response to complex environmental changes, that it kind of requires a whole set of strategies. And it's also that we're working, which I think is really powerful, that we're working with wild sampled populations. So they have all the tools, as opposed to kind of lab inbred lines that have lost, they're powerfully maintained, like isogenic lines, so you can really figure out genetic mechanism, but there's less fuel or fodder for evolution to work with. So I think by working with this standing genetic variation, we were able to reveal all of these different genetic mechanisms and epigenetic mechanisms, and see that there's a lot that's important. And we actually, we had a lot really strong signatures of functional enrichment with the alleles, but not with the epigenetic variation, which I don't know if that's just that we don't have enough. It was still like 100,000 loci, but I don't know. I don't know why we don't have any functional enrichment there, but we don't. So maybe it's just across the board, everything needs a little methylation.
Okay, I think we'll stop there and